in previous lecture we studied about the important terminologies which are involved in the genetics, and in today's lecture we'll start with classical genetics, right? So lecture 2 is about Mendelism, which is also known as classical genetics. So classical genetics is the oldest discipline of the genetics, where we will go back and study the experiments of this man, Gregor John Mendel, right? So Gregor John Mendel is the father of genetics. He was an Australian monk. He studied the inheritance of traits in pea plant. There's a reason why he selected pea plants. We'll discuss this later on. So he is the man who made it possible to identify the basic mechanism of heredity. So he established the rule of heredity, which are referred as law of inheritance. We'll also see what are the different laws of inheritance. His work was ignored until the turn of 20th century. So Gregor John Mendel, he got famous, or his work got acknowledged posthumously. Posthumously means after death. So his work was acknowledged by 3 scientists. Mendel's work was rediscovered by Carl Correns, Chechmark and Hugo de Veres. So these 3 scientists, they worked independently. They helped to expand the awareness. They helped to expand the awareness of the Mendel's law of inheritance. Mendel's law of inheritance. So the next point is that why Mendel selected the pea plant. What are the advantages of pea plant? So Mendel selected this pea plant for hybridization, right? So hybridization is the process of crossing two genetically dissimilar plants. So dissimilar plants, right? Say for example, Mendel wants to cross this pea plant with another pea plant. It's a Mendel pea plant. So these are 2 different varieties of the pea plant, right? So this is known as hybridization. So the reason why pea plant was selected: 1, this crop matures in 60 to 70 days. So this crop has very short duration time. The results are visible after 2 to 3 months, right? Secondly, pea plant has hermaphrodite flowers. So it's very easy to conduct hybridization, right? So it's easy to do self-pollination, and it's easy to conduct cross-pollination. So cross-pollination, self-pollination are the aspects of the hybridization. So what are hermaphrodite flowers? Hermaphrodite flowers are the flowers which has both reproductive structures. It has female reproductive structure as well as male reproductive structure present within the same flower. So these are both the sexes in the same flower. So these are hermaphrodite flowers, right? Say for example, if Mendel was interested to do self-pollination, so he will seal this flower upon maturity. So automatically this flower will self-pollinate, right? The anther of this flower will the pollens of this flower will fall on the stigma of the same flower, right? If Mendel was interested to do cross-pollination, he wanted to cross these 2 different varieties of pea plants. So what he will do, what he does, he will remove the anthers from this plant, because he's treating this plant as a female plant, right? So this process is known as emasculation. Removal of anther from a flower is known as emasculation. So he will emasculate this variety of the pea plant, because he is considering this plant as a female plant, right? Now he will collect the pollens from some other plant. So he'll collect the pollens of this pea plant, because he's considering this as a male pea plant. So he will collect the pollens and he will he dusted the pollens onto the stigma of this flower, right? The green variety. So this is how cross pollination or cross breeding is done. It's very simple to do self pollination and cross pollination or hybridization in such kind of flowers. Thirdly, pea plant has many contrasting characters. There are varieties which show round seeds, their seed color is yellow, their pod fruit is inflated, and their fruit, the pod color is green, right? In contrast to, there are varieties with wrinkled seed forms, green seed colors, pod is constricted, pod is yellow in color. So it was easy for the Mendel to crossbreed such kind of varieties and to see the outcome of these hybridizations, right? So pea plant has many contrasting characters to study. So this is again a reason why pea plant was selected. Fourth reason was small genome size. Pea plant is a diploid organism. It's a diploid plant, right? In previous lecture I told you about diploid organism. So if you pick a cell of pea plant, so it has total 14 chromosomes. It has total 14 chromosomes. So each chromosome is present in pair. This is a pair of chromosome 1, right? So this is the pair of chromosome 2. This is the pair of chromosome 3, and so on. So in total, pea plant has 7 pairs of chromosomes, right? So these pair of chromosomes, you already know, are known as homologous chromosome. So the genome size was very small. It had only 14 chromosomes. This picture is basically showing a cell with only single set of chromosomes. So that's why it has only 7 chromosomes. So such picture you can call these chromosomes as haploid set of chromosomes. So this is haploid number of chromosomes in a pea plant. Right, so this is the haploid number of chromosome in a pea plant, but pea plant is a diploid plant. So in total it has 14 chromosomes. That means 7 pair of chromosomes. So the different characters which were selected by Mendel, say stem height, flower position, then pod color, they were present on chromosome 4. Right, so they were present on this chromosome 4. Then flower color, pod color, right? So flower color, the genes for the flower color, they were present on chromosome 1. Then for pod color, they were present on chromosome 5. Right, so seed shape was controlled by the genes which were present on chromosome 7, and seed color was controlled by the gene which was present in chromosome 1. So only chromosome 1, 4, 5 and 7. So all the set of 7 traits, all the set of genes which were controlling 7 traits in pea plant, they were present on 4 chromosomes, only 4 chromosomes. So one thing I want to make clear is the character and traits. I'm using these terms again and again. So what's the difference between character and trait? So character is height, the stem height is the character here. So height can be of two types, either the plants they were tall or the plants were the dwarf. So tall and dwarf are the two traits of a character. Flower color can be either purple or flower color can be white. So flower color is the character. So the two traits for the flower color are purple color flower and white color flower. Flower position is a character. So the flower they can be present at terminal region or flower can be present at axillary region. So the axillary and terminal positions are the two traits for flower position and so on. So this is the difference between character and the traits. Well, now we will discuss about the monohybrid cross. Hybrid is when the alleles are of different kind. So this is a hybrid, right? Now this is monohybrid, means that is a single hybrid. So we are considering only a single pair of gene. We are considering only a single trait, or you can say we are considering only a single character. So that character is height. So height is the character. It has two traits. Either the plants are tall or the plants are dwarf. Right? So that's why it is known as monohybrid cross. So Mendel selected two types of plants. One was tall, second was dwarf. And then he crossbred these two varieties. Right? So this is the parental generation, P generation. So this is parental generation. Right? So then he collected after 3 months, as you know the crop matures in 3 months, he collected the seeds from the female plant. He collected the seeds from this plant, then sown the seeds into different rows, and what he noticed, he found all the plants in the next generation, they were tall. All the plants, they were tall. They were only this kind. Right? So they all were tall in the F1 generation. This was F1 generation. So then what he did, he self-pollinated the F1 generation. He collected the pollens of this plant and dusted on the stigma of the same plant. So what do you call it as? He did selfing. And then there was a seed set on this plant. He took the seeds, he sown the seeds into different rows or into multiple rows, and the next generation plants were of different kind. So he found in the next generation, in F2 generation, this was the F2 generation, some plants were tall. Some were dwarf, some were again tall. Yeah. So he found two kinds of plant, tall plants as well as dwarf plant in the population. Right. So Mendel was, we will see the mechanism behind it. Mendel was doing all this experiment on the basis of phenotype. He was just observing the external appearance, right? So he was selecting the plants on the basis of phenotype, on the basis of height. He was not aware of the genes. He was not aware of these factors. So Mendel knew nothing about genes. This concept was later on discovered. Mendel called gene as factors. right? So he described this whole mechanism as that there are certain factors in this plant and in this plant which were combined together, and these plants were tall. Upon selfing, there were certain factors because of which these plants were tall and this plant was dwarf, right? So later on, it was found that these factors are basically the genes, the pair of genes which were passing from one generation to the other. So now we will see the mechanism behind the monohybrid cross. So Mendel took a tall plant, tall parent, whose genotype was homozygous dominant, and he mated it with a dwarf parent whose genotype was homozygous recessive. So during gamete formation, as you know, during cell division, during gamete formation, the homologous pair, it separates. So this member will go to one gamete, the other member of the homologous chromosome will go to the other gamete. Similarly, the dwarf plant will produce two types of gamete. So this is the segregation of the chromosomes during gamete formation. So this is the rule. Because of that rule, the segregation of chromosome, so the gamete formation will take place. Now these gametes, they will fuse together. They will fuse together during the event of fertilization. So there is fertilization taking place here, right? So in F1 generation, if this gamete fuses with this, or this gamete fuses with this male gamete, this gamete fuses with this or this, so there is possibility of formation of only this kind of individual. So you can write female gametes on the top and male gametes on this side, right? So the possible offsprings will be, or the genotypes will be hybrid. They will be heterozygous, right? So they are heterozygous. The F1 individuals are heterozygous. Heterozygous are also known as hybrid. So why they are known as monohybrid? Because they are hybrid for one pair of allele. If the condition is like this, if an individual is hybrid for two different pair of alleles, then you will call it as dihybrid. We'll discuss dihybrid in next topic, under next topic. So right now it's a monohybrid because it is a hybrid for one pair of allele, right? So after F1 generation, Mendel did selfing. Now again you will draw the gametes. So the kind of gametes produced are, there are two different kind of gametes. You will draw the Punnett square to find out the possible combinations of the offsprings. So you will write male gamete, female gametes on the top and male gametes on this side. And if these two gametes, they fuse together, and if this and this gamete fuse together, so there is possibility of formation of a hybrid. So there is possibility of these kinds of offsprings, right? Now we'll look at the phenotypic ratio. So the phenotypic ratio in case of monohybrid cross will be, this individual will appear tall, this individual will also appear tall, this will also appear tall, but this one will be dwarf. So the phenotypic ratio is 3:1. And if you look at genotypic ratio, if you want to find out genotypic ratio, you want to see the genetic constitution. So you have one individual who is homozygous dominant, you have two individuals who are hybrid, and you have one individual who is homozygous recessive. So genotypic ratio comes out to be 1:2:1. Now I'll explain why this hybrid will be tall, right? So this individual has two alleles. Dominant allele is the allele which can express. This allele will produce sufficient amount of protein or a factor which is responsible for tallness in plants. So in heterozygous condition, the allele which express itself is known as dominant allele. So these hybrids are tall because of the presence of this dominant allele, right? The another allele which is not able to show its expression in the presence of dominant allele is known as recessive allele, right? So in case of homozygous dominant, again this individual has dominant allele which can produce, rather it has two dominant alleles. So these alleles were able to produce the protein, sufficient protein, which was responsible for tallness in plants, right? But in case of homozygous recessive protein, there was no dominant allele, so no protein or not sufficient amount of protein was produced which can cause the tallness. So there was no tallness. The plants were dwarf. Now we will study about the back crosses. So what are back cross? Back cross is when a hybrid, this hybrid is from F1 generation, is crossed with any of the parent. When it is crossed with any of the parent, either with the dominant homozygous parent or recessive homozygous parent. So this type of cross is known as back cross, right? So these two individuals, they are the parents. So you are going back, you are crossing the individual, you are going back and crossing the individual with any of the parent is the back cross. Second type of experimental cross is the test cross, right? So test cross is a type of back cross where you cross the F1 hybrid particularly with recessive parent, right? So this type of experimental cross is when the F1 hybrid is crossed with recessive parent. So what kind of genotypes are possible in this case? If you cross hybrid with recessive parent, the gametes will be written on this side, the parental gametes will be written on upper side. So there is the possibility of two kind of individuals. These two individuals will be tall and these two individuals will be dwarf. So tall and dwarf, they are in the ratio of 1:1, right? So this is the phenotypic ratio, as well as genotypic ratio is also same as phenotypic ratio. Genotypic ratio will, again, there are two types of genotype possibles. So they are in ratio 2:2, that means they are in ratio of 1:1. So genotypic and phenotypic ratios are same in case of test cross. So what's the importance, significance of the test cross? You have a plant which is dominant. You have a plant which is dominant, but you are not sure about the genotype, whether the plant is tall because of this genetic makeup or the plant is tall because of heterozygous condition. So you want to find out the genotype of an individual, right? So in this case, if the individual, we will consider this as first case. In this case, the possibility will be, if you draw the Punnett square, cross it with the recessive parent, the possibility will be all individuals produced in such kind of cross will be tall. All the individuals will be tall. And in this case, case B, if you cross the plant with the recessive parent, the possibility will be you'll get what? You'll get tall plants as well as dwarf plant. So on the basis of this, you can decipher the genotype of these individuals. Right? The third kind of cross is the reciprocal cross. The third type of experimental cross is the reciprocal cross. Suppose there's a plant, pink color plant, is crossed with another plant which is blue in color. Right? So in the first experiment, this is considered as female and the blue color plant is considered as a male. Then in the second experimental cross, you are switching the genders. Now you are considering blue plant as female plant and you'll consider pink plant as the male plant. So in case of reciprocal crosses, the sexes are interchanged in the two cross. Right? So what's the importance of reciprocal cross? If you want to see the effect of gender or sex on a trait. So in next type of cross, dihybrid cross, Mendel considered two characters. Mendel considered two characters together. One character was the shape of the seeds and the second character was the color of the seeds. And very obviously the shape of the seeds, they were controlled by a different pair of alleles, either RR or R small r or small r, and the color of seeds was controlled by another pair of allele, right? Either homozygous dominant, either hybrid or recessive. Fine, so this was located on chromosome 7. These pair of alleles, they were located on chromosome 1. So the two characters, they were considered together in dihybrid cross. Now, Mendel selected a female plant which was having these two characters, these two traits. The seeds were round in shape and the color was yellow. The genotype of the female plant was homozygous dominant for both the traits, and for the male plant the genotype was homozygous recessive for both the traits, right? So he did a cross between these two pure parental plants, right? So what kind of gametes are produced by female plant? There's a possibility of formation of only one kind of gametes, right? And in case of male plants, there's a possibility of formation of again one kind, different but one kind of gametes. So these are the gametes which are produced in the parental generation. Now we will find out the possibility of formation of the genotypes, right? So if you write the gametes on this side and male gametes on the upper side, so there's a possibility that all plants which are produced in the F1 generation, they are hybrid. There is no other possibility. All have same genotype. They are hybrids. So why we are calling this plant as dihybrid? What's the reason? Because it is a hybrid. It's a hybrid for both the pair of alleles. It is hybrid for this as well as for this pair of alleles. So this is a dihybrid. If you remember, in case of monohybrid, the condition was this only. right? You are just looking at one pair of allele. When the one pair of allele is present in a heterozygous condition, then it is monohybrid. Here both different pair of alleles, they are in heterozygous condition. So that's why you are calling it as a dihybrid cross. So all the individuals which are produced in F1 generation, they were dihybrids. Now what Mendel did in F1 generation? He did selfing. It was crossed with the same plant. Now what is the possibility of gametes now? We'll see what kind of gametes are produced in F1 generation. So if the parent has this genotype, so you can split it this way, right? So this is how we make the gametes. The first possibility is this. The second possibility is, third possibility is small r, capital Y. Fourth possibility is small r, small y. So these are 4 types of gametes which will be produced in F1 generation. Similarly, this will also produce these 4 kind of gametes, right? So now we will draw the Punnett square, right? So it will be a Punnett square with 16 boxes. So there are 16 possibilities. You'll write the gametes, female gametes on this side, and male gametes on upper side. Right? Now, if this gamete fuses with this male gamete, the possibility of the zygote will be homozygous dominant. Next zygote will be this kind, right? So I'll draw all the possible genotypes here. So this was a huge cross. So there is possibility of 16 genotypes, right? So the first zygote, this will have what kind of traits? So this will be, we'll write over here, round yellow. It will have round seeds and the color of the seeds will be yellow. So this is round yellow, right? So we'll mark it with yellow color for convenience. Next, this is round yellow. Next, again, we have a dominant allele here, so it will be round. We have a dominant allele for the yellow color, so it will be round yellow. This will also be round yellow, round yellow. Again, it has a dominant allele. It has dominant allele for color as well, so it will be round yellow. This, just look at this. This will be round, but it doesn't have any dominant allele for the color. So it will be green. The color of the seed will be green, right? Now, this is round green. This is round yellow. Again, this will be round green. Fine. This will be round yellow. Again, this will be round yellow. And next is, this will be round yellow. Right. So, 1, 2, 3, 4, 5, 6, 7, 8, 9. So, total round yellow kind of genotypes will be, not genotypes, sorry, the zygotes will be 9. Okay, so there will be 9. So how many round green? We'll find it out. So this was one round green. Then this is also round green. So we need, yes, so this is also round green. Why it is round green? It has a dominant allele for the shape, but it has no dominant allele for the color. So there are 3 round green type of individuals, right? So we'll look at this now. What will be the case with this? There is no dominant allele for roundness. So this seed will be wrinkled, and but there are dominant alleles for the yellow color. So they are yellow, wrinkled yellow. Fine. So one is wrinkled yellow. This is again wrinkled yellow. This is again wrinkled yellow. So there are 3 individuals which will have this type of phenotype. Fine. So what about the last one? So this is wrinkled again, and this will be green. So wrinkled green, there will be only 1. So what's the phenotypic ratio? The phenotypic ratio comes out to be 9:3:3:1. 9 and 1 are the parental type, and these 3, this and this, round green and wrinkled green, they are known as recombinants. They are recombinants. So if somebody asks you about the ratio of recombinants, you can find it out from these values, right? So originally these were not the parental traits. The parental traits were round yellow and wrinkled green. Now the trait roundness is combined with green. So the traits are recombined. So these are known as recombinants. Fine. So I repeat, the phenotypic ratio in case of dihybrid crosses 9:3:3:1. Now we will simply make a trihybrid genotype here. So this is a trihybrid, right? So I will show you how gametes are formed, right? How you can make the gametes, how you can find out the different kind of gametes which will be produced by this individual, right? So you split these 2 T's, write R in front of both. Then further you split it, write Y, small y. So this is the way you can make the combinations, right? So the first type of gamete produced in this case will be capital T. You'll read it this way: capital T, R and Y. Second kind of gamete, the possibility of second kind of gamete will be capital T, R, small y. Then capital T, small r, capital Y. I mean this thing, right? Then fourth possibility is capital T, small r, small y. This will produce small t, capital R, capital Y, small t, capital R, small y, small t, capital R, capital Y, small t, small r, small y. So these are different kinds of gametes. So they are basically eight different kinds of gamete, 8 different kind of gametes which are produced in a dihybrid individual. So next are the Mendel's law of inheritance. Mendel laid down certain rules of inheritance which are referred as Mendel's law of inheritance. The first law is law of dominance. What it says: when parents with pure contrasting traits are crossed together, say for example, you take a parent who is pure tall, homozygous tall, You cross it with a pure parent which is recessive, which is DOF. So parents which are pure and they have contrasting traits, they are crossed together. Only one form of trait appears in the next generation. So what will be the outcome in the F1 generation? All individuals, they will be hybrid. So only one form of trait appears here. And what will appear here? It will be, the hybrid will exhibit only the dominant trait. So what's dominant in this? T is dominant allele. T is dominant allele, right? So only T would be able to express. The another allele, which is the recessive allele, it won't be able to express along with the dominant allele. Recessive alleles express only in homozygous recessive state, right? So in case of hybrid, only the dominant allele will express. This allele will produce the sufficient amount of protein which will be responsible for the tallness in the plant, right? So this is law of dominance. So next, second law is the law of purity of gametes, which is also known as law of segregation. So this law is basically explaining the segregation of chromosomes, or you can say the segregation of alleles during cell division. So consider this a cell which has one homologous pair. I'll draw another homologous pair as well. So this is another homologous pair. So the alleles present over here are, this will consider a different pair on this chromosome, right? Now you just focus on this pair of allele, right? So basically you are studying here intra-allelic segregation. Intra-allelic segregation, you have to focus on this, right? So during gamete formation, this cell will produce two cells by meiosis, right? So both these cells, they will have half number of chromosomes. So these are basically the gametes. The gametes will have half number of chromosomes. So now you consider only this pair of alleles. So both these alleles, they will segregate independent of each other. One T will go in one gamete, another allele will go in another gamete, right? Now you consider the second pair of allele. One will go in one gamete, another will go in another gamete. There is no such relation that both the alleles, they together go into same gametes. So such kinds of gametes are not possible. By rule, this pair of allele, it has to segregate independently of each other, right? So one chromosome will go to this, the another member will go to this. So the third is the law of independent assortment. Third law is the law of independent assortment. First and the second law, they were deduced on the basis of monohybrid cross. Now this is the third law. Again we will see how the homologous pair, they segregate during gamete formation. We'll draw a pair of alleles here and also here, right? Now you will see the relation between this pair of allele with different pair of allele, right? So now you have to focus on inter-allelic segregation during gamete formation, right? So what kind of gametes are, possibilities of gametes formation will be there? right? So this T will assort independently from this T, very true. This R will independently assort from this R, but there is also a possibility that the small r goes with capital T, right? And there's also a possibility that the small t goes with capital R. So there is the possibility of formation of 4 kind of gametes. So I'll repeat, this R is independent of segregation of this T and this T, right? So there is no such rule, there's no such relation that this R says that it will go only along with capital T, right? So it's not like there will be possibility of formation of only this kind of gametes. No, R can segregate with capital T as well as R can go with along with small t. So this is law of independent assortment, which was this law is deduced on the basis of dihybrid cross. Third law is deduced on the basis of dihybrid cross. Why dihybrid? Because you are considering here two different set of alleles. Second important thing under this law is that this law is not universally true. This law is, the third law is not universally true. It is applicable to only the pair of alleles which are present on different chromosomes. So these pair of alleles, they are present on different set of chromosomes. Or it's also applicable to the alleles which are present on same pair of chromosome, but they have a huge distance, right? It's not applicable to, this third law is not applicable to the pair of alleles which are present on same chromosomes and they are very close enough. If the distance between these pair is very small, then this law is not applicable on these different pair of alleles. No, these setup of alleles, because in this case the segregation, this R will always segregate along with capital T because they are present on same chromosome. So they will segregate this way only, right? And these alleles, they will segregate together, right? So now we will practice some question on Mendelism, right? So which of the following is the monohybrid ratio? So you are well aware that when Mendel crossed two pure plants, pure tall and pure dwarf, the F1 generation were all tall and they were hybrids. And when selfing was done, there was possibility of 4 or these kinds of individuals. So the phenotypic ratio was 3:1. These 3 individuals were tall and this was dwarf. So the ratio is 3:1. Purity of gametes. So this is the second law which was deduced on the basis of. So this was deduced on the basis of monohybrid cross, because in purity of gametes we are considering only one pair of allele, right? We are looking at the segregation of only these 2 alleles, right? So only one pair of allele is considered here. So it is, you can say it was deduced on the basis of monohybrid cross. Then there is a cross hybrid. It's a kind of test cross, it looks like it is crossed, right? It is crossed with the recessive genotype. So what will be the F2 generation? Fine. So you draw all the possibilities here. So what we find in this? So the individuals, they are tall, and there are individuals who are dwarf. So the ratio is 1:1. So tall and dwarf in the ratio of 3:1, which is wrong. Are they all tall? No. Again, this is wrong. All dwarf? No. So tall and dwarf are in the ratio of 1:1. So the answer is none of above. A gene is said to be dominant. A gene is said to be dominant if it expresses in homozygous state, also when it expresses in the heterozygous state, right? So the answer is that it never expresses. This is wrong. It expresses only in heterozygous condition. That's wrong. Yes, it do express in heterozygous condition, but not only in heterozygous condition. So it expresses in homozygous condition. Recessive alleles, they are able to express only in homozygous conditions. They are not able to express along with the dominant allele. So a gene is said to be dominant if it expresses both in heterozygous condition and homozygous condition. So answer is D. A cross where sources of gametes are reversed. So this kind of cross is reciprocal cross. Like in first cross, you are considering this plant. Okay, we'll mark it as A and B. A and B plant. A is female and B is male. And in second cross, you are considering B as female and A as male. So the answer is the type of crosses where genders are reversed, they are known as reciprocal crosses. So the next question is, they are asking about a cross, the possibility of a cross. So R is dominant over small r. R produces red pigment. So the F1 generation will consist of all hybrids. So as per the information given in the question, R is dominant allele which produces red color. So all the progenies in F1 generation, they will be hybrid. So all individuals will be red. Well, the next question is, how many types of gametes will be produced in F2 generation of a dihybrid cross? So in F1 generation, the dihybrid is this kind, right? So you have to produce the types of gametes here, right? So I guess it will come out to be, it will produce 4, how many? It will produce 4 different kinds of gametes. And it's not in F2 generation, this is in F1 generation. The question should be F1 generation, right? So answer is 4. Now next question is, white flower W is dominant over black small w. 2 white flowers are crossed to produce 9 white and 2 black flower. So this, what are the genotypes of the parent? So the ratio is 9:2, which is very much near to 3:1. So the answer seems to be a cross like this. Cross like this, right? But we'll rule out each and every option. So in this case, there is no possibility of production of white flowers. So this can't be the answer. In this case, there is no possibility of the formation of black flower because you need this allele for the production of black flower. So in this case, if you try to find out the possibilities of the genotypes, so what kind of individuals will be produced? If you consider this cross, it will be 50%. This is 50% white, and again, no, it's all 100% white. There will be no black flower because these are hybrids. Again, the dominant allele will express, right? So the only possible answer comes out to be A. Ninth is, what is the ratio of true breeding tall to true breeding dwarf in F2 of monohybrid cross? Now we have to draw the possible genotypes in F2 generation, right? So these are the possibilities of zygote in the F2 generation. So the pure tall are 1, these are hybrids, so you will not consider them, and pure dwarf are 1, so the ratio is 1:1. So T is to this. 2 pea plants, they were subjected to cross pollination. The next question is, 2 pea plants, they were subjected to cross pollination. Of 183 plants produced in the next generation, 94 plants were found to be tall, 89 were dwarf. You have to find out the genotypes. Again, if you look at the ratio, this is very close to the ratio of 1:1. So it has to be a test cross. Test cross is where the hybrid is crossed with recessive parent. So answer seems to be very clearly, answer seems to be C. But again, we'll go by each and every question, right? Every option. So in this case, as there is no allele for dwarfness, there is no recessive allele, so no dwarf plants are possible. So this can't be the answer. In this case, if you try to find out the possible genotypes, so all the plants, most of the plants, they will be tall, so the ratio will come out to be 3:1, right? So your plants, they are not following the ratio of 3:1. So in this case, it doesn't have a dominant allele, so no tall plants are possible. Again, we'll rule out this option. So the answer is this. The total number of dihybrids produced in F2 generation. You have to find out the dihybrids. This is a very lengthy question, right? So you have to make all the possible 16 genotypes over here. Fine. So we'll see whatever the different kinds of gametes they were produced. So it were this, and this. Again, this, and this. Now, fuse these two gametes. Write all possible genotypes here. Right, so you have to find out dihybrids in this. So let me complete this Punnett square. So it's a very important question. This question was asked in Junior Science Olympiad. So the answer will come out to be, so they want to know the total number of dihybrids. Do dihybrids mean hybrid for both the allele? It should appear like this. Genotype should be like this. It should be hybrid for this pair of allele as well as it should be hybrid for heterozygous for this pair of allele. So I can see this is one dihybrid. This is not dihybrid. It is hybrid for only one pair of allele. Right? If you look at the second pair of allele, they are homozygous, dominant. It's not hybrid. So this is one dihybrid. This is second dihybrid. Well, this is third dihybrid and this is fourth dihybrid. So the answer comes out to be 4. Right, so in the F2 generation, there are formation of 4 dihybrid, these kinds of genotypes. The genotype of, next question is, the genotype of dominant parent is determined by crossing it with a recessive parent. So this type of cross is the test cross. In test cross, if you are not sure about, you selected two tall plants and you are not sure about their genotype, whether it is homozygous dominant or it is tall because of heterozygous condition. So what you will do, you will cross it with the recessive parent, right? So in this case, there is no possibility of dwarf plant, but if the selected plant dominant parent is heterozygous, so there will be possibility of both tall and dwarf, right? So you can find out, you can decipher the genotype of the dominant parent here, right? So the answer is the test cross.